Thank you for this opportunity today to speak about COVID-19 vaccines. I have no conflicts of interest to disclose. All vaccine development begins with a look at the pathogen. So this is a picture of SARS-CoV-2, which is a coronavirus. The viral particle, which contains RNA inside, is coated on its surface with spike proteins, and these are shown here in red. This is an essential protein for viral function because it binds to the ACE2 receptor and mediates entry into the cell. On the right is the spike protein. It's a structure. This was resolved in March. And this picture is showing that the spike protein is rooted in the membrane of the virus. And at the very top of the protein, what is seen most prominently by the immune system is shown in green, and that's the receptor binding domain. So there are all types of protective immune responses that are generated after exposure to a pathogen or to a vaccine. And usually for viruses, the first thing that we look for are neutralizing antibody responses. So these are antibodies that bind and inactivate the virus, and they actually prevent viral entry into cells from the beginning. They can also promote viral clearance. We do measure other things like binding antibodies that don't have a lot of function, or non-neutralizing antibodies that have different effector functions. And also, of course, cellular immune responses, which are required for coordination and for CD8 positive T cells to actually kill infected cells. Well, what do we know about natural immune responses to SARS-CoV-2? And I've underlined responses to say instead of immunity, because we actually don't know yet if humans can generate immunity to SARS-CoV-2, either by infection or by vaccination. But this study came out last week. This is from investigators at Rockefeller. And in this study, 149 individuals who had recovered from COVID-19 were analyzed, serum were analyzed. And what they found was that there was a wide range in neutralizing antibody titers, and that's shown on the right. So that's, in that figure, they're showing the distribution of the titers in both outpatients and in a smaller group of individuals who were hospitalized, so quite symptomatic. In regard to that wide range, there were a lot of people who actually had pretty low titers, but also some that had extremely high titers over say 5,000. And what they found by looking at these two different populations is that the magnitude of the antibody response seemed to correlate with severity of disease. And that's shown in the fact that the hospitalized patients had significantly higher titers. Overall, the geometric mean titer was about 121. So in their analysis, they also found that antibodies were very similar among the different participants. They all targeted spike protein. They even targeted the same domains, like the receptor binding domain. And even if it was at low levels, they were able to identify potent neutralizing antibodies in pretty much everyone that they looked at. And that's quite supportive of a vaccine, because that means that a vaccine might be able to elicit potent antibodies in nearly everyone. But can you get immunity to SARS-CoV-2? And that's a question that can be looked at in non-human primates. So this is a study that I'm going to show from my colleagues at the Center for Virology and Vaccine Research under the leadership of Dr. Dan Baruch. In this experiment, 9 rhesus macaques were infected with SARS-CoV-2 via the upper airway. And all of the animals in this animal model had high viral loads in BAL, or bronchoalveolar lavage. So that's fluid from the lower respiratory tract. And that's shown in the figure to the right, looking at that panel of first challenge. There, the monkeys had a peak in their viral load around 2 days, and then a slow clearance of the virus that corresponded to some symptoms in these monkeys, taking up to 2 to 3 weeks to fully clear. These animals were similar to the humans. Once they recovered, they had median neutralizing antibody titers of about 100. So at day 35, these animals were re-challenged, again with SARS-CoV-2 via the upper airway again. And that's shown in the panel to the right called second challenge. In that panel, you can see that a handful of animals had a blip in their virus that was detected in the first couple of days, and then rapid clearance. And there was no recovery of actual live virus from these animals, and most of the animals didn't have any detectable virus in their BAL, though a lot did in the upper airway, which is kind of interesting. Nevertheless, this is a picture of some kind of immunity. So they certainly looked to be protected from reinfection. Not sterilizing immunity, because a lot of them did have a blip of virus, but at least rapid clearance. And that was also supported by the fact that all of the animals had a rapid boost in their immune response. And that shows immune memory. Well, can you elicit this type of immunity with a vaccine? So this is another study that came out of our group at CBVR, in which 6 DNA vaccines were designed, all of them looking at that very important spike protein. The difference between these different DNA vaccines is that the first one, the S vaccine, encoded for the full viral protein all the way through the transmembrane domain, which anchors it to the virus in the little cytoplasmic tail, to other versions that just included the receptor binding domain, essentially like a very long peptide, which is shown in green at the top of the figure. So after immunizing these monkeys, they found that 2 of the DNA vaccines were effective at eliciting neutralizing antibody titers, and in this case, actually higher than 100. And these vaccines were similar to each other. They were essentially the full length of the spike protein. The only difference was that one had chopped off the cytoplasmic tail, but they all had that anchoring transmembrane domain, which it seems to be important for the structure and maintaining the structure and the presentation of the receptor binding domain. So 4 of the DNA vaccines, the ones that were all progressively more truncated, were actually really poorly immunogenic. So this was helpful to know that you're not going to get away probably with a little peptide vaccine. You're going to need the full protein. Antibodies were specific to the spike protein as predicted, and also to the receptor binding domain. So at week 6, all of the animals were challenged via the upper airway, and that's shown in the figure to the right. The panel of placebo show the animals challenged with placebo that had the same kind of kinetics. They had a high viral load in their BAL, peaking around day 2, and then taking a couple weeks to fully clear. Animals that got that more effective vaccine in terms of antibodies, the spike vaccine, and that's shown on the right, had a blip in their viral load and then rapid clearance, very, very similar to the convalescent monkeys. And a subset of animals, about 8 of them, actually never had any detectable subgenomic RNA, which is a measure of actively replicating virus. Importantly in this study, and I think really critical for going forward in clinical trials, is that there was an inverse correlation between the neutralizing antibody titer and who was protected among these monkeys. And that's shown in the figure on the bottom, neutralizing antibody titers versus viral load in the scatter plot, which shows a significant relationship. And to my eye, when I drop down a blue line there for the titer of 100, I see a kind of natural breakpoint, and this is not sophisticated, but above 100, most of the animals were protected, and below 100, almost all of them had high viral load. So it's kind of a biomarker that you can use in vaccine development. It may even be the mechanism of protection. What kind of vaccines do we have in that clinical pipeline? Well, there are many, many different types of COVID-19 vaccines. What's important to really know is that, I mean, they're all using the spike protein, because that seems to be a very critical component. So it's all the same spike protein, full length, just different mechanisms of delivery. And that could be by an inactivated virus, just the spike protein, or different methods of delivering the genetic code for the spike protein. So DNA plasmid, messenger RNA, or a viral vector. On the New York Times, you may have seen there's a really nice coronavirus vaccine tracker, and it's fun to keep an eye on all of the different things coming forward, but it can also be overwhelming to look at, more than 100 preclinical constructs in development. So what I want to do is just focus in on what's actually made it into clinical trials and moving forward into phase 2 or phase 3, and that also have some data to share with you. Now, vaccine development is occurring in a very unusual paradigm, which was referred to in the New England Journal as the outbreak paradigm of vaccine development. And what's unique about this paradigm is that the phases of development are overlapping, as opposed to being in sequence. And that's really the key difference. In addition, it's also multiple vaccines going into phase 3 efficacy studies instead of, say, 1 or 2 of the most promising. So in this figure, we have, hopefully, and the way it's intended to be, is that all of the same critical safety assessments are occurring and with the same rigor as they would be otherwise. But what is different is that what's happening at the same time is that we're having global scale manufacturing, because the vaccine has to be safe, effective, and deployable in order to be useful in an outbreak. And that's shown here on the bottom, that the manufacturing is happening now, even though we only have a little bit of phase 1 data. And that is going to involve a tremendous financial investment by governments, by companies, and that's really what makes this a different paradigm. So just to focus on a handful of the vaccines where there's data, and I should say I won't be able to share much description of other vaccines, like even some that we're working on in our group that are quite promising, and I'll just have to save that for a future talk in which we do have more data on them. So the first vaccines with data actually came out of China, which is not surprising given that they were the first to really experience this outbreak. This is a company called Sinovac, which created an inactivated virus vaccine. And this is an old-fashioned but tried and true type of vaccine technology. So basically what you do is you kill the virus and then you purify it through multiple different phases of purification. And the trick is to kill a virus without destroying its structure, because you want it to look like the real thing. So they were successfully able to do that when they, and you can see that in that electron micrograph, where they show viral particles that looked intact, and you can even see the spike proteins on the surface. So they injected this inactivated vaccine into monkeys and other types of animals, found again that they elicited nice neutralizing antibody titers up to about 100. And when they challenged these monkeys, they found a significant reduction in viral RNA in lungs and in various different swabs. And what's shown on the right is the throat swab, so upper airway. And you can see that in the vaccinated monkeys shown in pink, they have a little bit of a blip, again, of virus in the upper airway, but then that is rapidly cleared over a couple of days. Importantly, they found in this study and described no antibody-dependent enhancement. So there's always a worry that your vaccine is going to somehow cause even worse symptoms and not protect. That's something that can be seen, say, in natural infection with dengue virus, but that was not observed here. And this vaccine is now in phase 1 and phase 2 studies. Now, per the media last week, the Sinovac company reported that neutralizing antibodies were elicited in more than 90% of the participants and that there were no safety issues, but take that with a grain of salt. That was purely in the press. Interestingly, the only vaccine study in humans to be published is a study of an adenovirus 5 vector vaccine. This is the very first that's out there, and this was in Lancet. This company, also a Chinese company, performed this trial. It's quite a nice study. And this was in collaboration with other Chinese groups. It was an open label dose escalation trial of 108 participants in Wuhan, China. And here they tested a replication defective adenovirus serotype 5 vaccine that encoded for the spike protein. They gave a single injection and then they followed participants for a month. So this is very short-term data. Now, to my eye, this vaccine elicited pretty low, moderate immune responses. So looking at the table, I've highlighted in blue the geometric mean titers observed with the different dose groups. And the highest dose group did elicit the highest titers, significantly better than the others, but the GMT was only about 34. So that's less than what we've seen in those animal studies or the convalescent plasma studies. In addition, the high dose was not particularly well tolerated. So a few patients had high fevers, muscle aches. And so they're actually moving forward into phase 2 studies without that high dose. I should add that what might be playing a role here and why these immune responses are lower than what was observed in the other studies I mentioned is that a lot of the participants had pre-existing immunity to Ad5 itself. So they'd been infected with adenovirus 5 somewhere along the way in their life, and that actually dampens the immune response when that happens. So perhaps the most widely known vaccine is the Moderna mRNA vaccine. And it's interesting that it's so widely known at this point, considering that it actually doesn't have a lot of data out there. Now, what it consists of, this vaccine, is a lipid nanoparticle that encapsulates messenger RNA that encodes for the full-length spike. And what the company really brings to the table is actually the nanoparticle, because the messenger RNA is very unstable and degrades quickly, and so you need a mechanism to keep it stable when you inject it. Now, they just recently put out in a preprint publication the results of some mouse studies. And in this one, they show in this figure here, the neutralizing antibody titers actually look fantastic in this mouse model. So the titers were about 1,000. And then they showed that when they challenged these monkeys with a mouse-adapted SARS-CoV-2 virus, again, they had a reduction in viral load, and it was really dose-dependent. So you can see this was related to the vaccine. So that's shown on the right-sided figure. So as you increase the dose, the protection became more pronounced. But they had less evidence of a change in the upper respiratory secretions. In any event, they again showed no disease enhancement. This is in mice, so this is not the greatest model here. Now, what made it newsworthy is that they really launched very quickly with this new technology, and they started their phase 1 trial back in March, and they tested a whole bunch of doses. They've added cohorts of patients, and now that phase 1 trial is actually up over 100 different participants. Now, they did release a press release, but it only reported some information about 8 participants. And what they reported in that press release is that the neutralizing antibody titers were similar to convalescent humans. Now, we can interpret that any way. We really don't know what that number is. As we've seen, that there's quite a wide range of titers in humans. And we also don't know why those 8 were presented and not others. Nevertheless, that is promising, what they've reported. And then in the media, it was also reported that that high dose, the 250 micrograms, that's the highest dose they tested, was also not very well tolerated with fevers and symptoms. And so the phase 2 trial, which is now ongoing, is testing 50 and 100 micrograms, excuse me. And the phase 3 trial for this is actually slated to begin in the United States next month. So this is a very rapid development of this vaccine. Finally, I'll end with just discussion of AstraZeneca, which is developed this vaccine in collaboration with Oxford. And this is an adenovirus vector vaccine based on a chimpanzee adenovirus. The advantage to this is that there's no pre-existing immunity among humans to this chimp adeno. So hopefully we won't have that issue of dampened immune responses. Again, it encodes for the full-length spike protein. So they tested this vaccine in non-human primates. Interesting, their titers are actually a little bit lower. Like you can see that in that figure, more like 50. But they did see some efficacy with it in the monkeys that they tested and challenged, where they saw vaccine prevented lung damage, the viral loads in the BAL were reduced. There wasn't any real change in the nasal swab RNA, but there's no disease enhancement. So a very similar kind of picture. They launched their phase 1 and 2 studies and are moving also quickly into phase 3 trials as they're doing global scale up of manufacturing. And so those studies may come out this summer as well in the United States. So to summarize, COVID-19 does lead to neutralizing antibody responses in humans, and that's very encouraging for vaccine development. There's a wide range in titers following infection with a GMT of about 100, and that's likely related to the severity of disease. Neutralizing antibodies target the spike protein, and it's currently unknown if those neutralizing antibodies in humans are actually protective against reinfection. That's unknown now. But it is shown now in monkeys, at least, that those convalescent neutralizing antibody titers are protective. Nearly all of the COVID-19 vaccine candidates that are in development use the spike protein. They all kind of use that same central piece. It's only the delivery mechanism that's really different. And finally, preclinical studies, they suggest that a successful COVID-19 vaccine is possible. So that is really encouraging. So vaccinated monkeys or animals using different platforms had reduced symptoms, they had reduced lung pathology, reduced viral RNA in many different compartments with rapid clearance. Now, sterilizing immunity is really not seen. That's where you have absolutely no evidence of virus after exposure, but you do see pretty rapid immune-mediated viral clearance with reduction of symptoms. Now, there's a new outbreak paradigm for vaccine development, and that is something we're going to have to keep our eye on how this goes. But multiple candidates are being tested at once, and global scale manufacturing is occurring simultaneously with the trials. Ultimately, a successful COVID-19 vaccine will need to be safe, effective, and deployable if it's going to make an impact on this pandemic. So thank you. I'm going to end there, and I will hand it back to our organizers.